Today we'll be talking about Mendel's laws of inheritance. And Mendel's laws of inheritance are based on the monohybrid cross that Mendel did. So all of these laws are taken as an inference from the monohybrid cross that he did. So just keep that in mind when you're thinking of all of these laws, that it is based on the monohybrid cross and nothing else. So coming to the laws itself, there are three laws. The first one is the law of unit factors, the second one is the law of dominance, and the third one is the law of segregation or the law of purity of gametes. So coming to the first law itself, the law of unit factors, what the law states is that any character that you find, any phenotype that you find, is controlled by a pair of factors. So there are two of them, so you have a pair. So a pair of factors control a character. A phenotype that you see is controlled by a pair of factors. And during gamete formation, so you know that gamete formation is occurring when the parents are going for fertilization. So during gamete formation, each gamete will get one from the pair. So pair meaning that you have two, right? And during gamete formation, one of them is going to get transferred to one gamete and another is going to get transferred to another gamete. And so the offspring is going to get one of the unit factors. So one from the pair is going to come from the mother, and the same will happen with the father, wherein there are two gametes which are being formed, and so one from the mother and one from the father, and these two will fertilize not in a not in a specific manner, but it's a more random way, and the offspring is going to get one from each, right? So the law of unit factors is stating that the phenotype, any phenotype, is governed by a pair of factors, and during gamete formation, one from the pair is going to enter each of the gamete, which is again is going to get passed on to the offspring. Now coming to the law of dominance, it is very self-explanatory that if you take a pair of factors, one of the factor is dominant as opposed to the other, which is recessive, which we have been talking about in monohybrid cross. So if you take a heterozygous tall plant, so a heterozygous tall plant would have a genotype of capital T and small t. Now phenotypically it is tall, but genotypically it has both the alleles for tallness and dwarfness. But because the tallness allele is a dominant allele, it is going to get expressed over the recessive allele, which is the dwarfness allele. So the law of dominance is saying that when you take a pair of alleles, one of them is dominant over the other, and that gets expressed every single time. Okay, so that is the law of dominance. Now when you come to law of segregation or purity of gametes, it's again going back to what unit factors is saying. So the law of unit factors and law of segregation is kind of similar in the sense that when a pair of factors are brought together, like in unit factors, when a pair of factors are brought together, the factors segregate or separate during gamete formation. So when you have a heterozygous tall plant which has a genotype capital T and small t, and this plant is going for gamete formation, you will have a separated gamete with the allele for tallness, and you will have a separated gamete with the allele for dwarfness. Okay, now there is a equation by which you can figure out what will be the F1 generation progeny, the frequency of the F1 generation and the frequency of the F2 generation. So if you want to figure out the frequency of the gamete, how often it is going to occur in either F1 generation or F2 generation, you can do it using the equation ax plus by whole square. Okay, so you can do it using the equation ax plus by whole square, wherein a is referring to one of the gametes, b is referring to another gamete. So if you take the example of crossing a pure breed tall plant with a pure breed dwarf plant, wherein in the F1 generation you'll be getting a heterozygous tall plant, in that case one of the gametes would be capital T and another gamete, B in this case, would be small t. Okay, and the x here refers to the frequency of T, meaning that how much of T is present, and y is referring to the frequency of small t, as in how much of small t is present. So if you take the F1 generation, T is half of it and small t is half of it, right? So you can see that the F1 generation is always made up of half of the tallness allele and half of the dwarfness allele. Now if you want to figure out what will be the frequency of capital T and small t in the F2 generation, you can do it by using this equation. So just insert everything that is there as a and x. So you have half capital T plus half small t whole square. So solving the equation, you would get half capital T plus half small t multiplied by half capital T plus half small t. And the answer, of course, would be 1 by 4 capital T plus half capital T small t plus 1 by 4th small t small t. Now if you look at the answer that you get, it is the same amount of ratio that you would get for the F2 generation, right? If you're going to self-cross F1 generation, wherein you're crossing heterozygous tall with heterozygous tall, the answer that you would get would be 1:2:1 for the genotypic ratio, which will be capital T is to heterozygous tall is to homozygous dwarf. Now this is the same answer that you would get here, but you also get the ratio of each of them occurring. So this is how you can use the equation ax plus by whole square. Okay. Now the next thing that we'll be talking about is backcross and test cross. Now the major difference between backcross and test cross is backcross is when you cross any kind of heterozygous genotype containing organism. In this case it will be mostly plant in case of Mendelian genetics, with a pure breeding plant. Okay, and backcrosses are always backcross can happen with any kind of genotype. So you can cross a heterozygous tall plant with a homozygous tall plant, or you can cross a heterozygous tall plant with a homozygous dwarf plant. Now this, any kind of cross where you're doing anything with anything which is its parent. So if you're crossing anything from the F1 generation with any of the parent, it's called as a backcross. But if you're doing a cross between F1 generation and the double recessive, keep in mind, if it is F1 generation being crossed with any parent, then it is a backcross. You can even do a F2 generation with the F1 generation, that would still be a backcross. But if you're doing a F1 generation with a double recessive parent, so it needs to be a double recessive parent, which in this example would be the double recessive homozygous dwarf plant. Then this kind of cross is called as a test cross. Now test cross is done to figure out the genotype of the F1 generation, because the F1 generation, although phenotypically is tall, is not genotypically homozygous, correct? So this is a heterozygous tall plant which is typically got in the F1 generation, but phenotypically it looks tall. It does not look any different from a pure breeding tall plant. So how do you figure out what is the genotype of the F1 generation? That you can do by doing a test cross, wherein you're crossing the heterozygous tall plant which you get in the F1 generation with its double recessive parent, which will be the homozygous dwarf plant. So in this case, the gametes that you will get will be capital T and small t crossed with small t and small t. Now if you draw a Punnett square, you have capital T, small t from the one of the F1, and then you have small t from the double recessive parent. So you would have half of them being tall and half of them being dwarf. So if you get an answer after crossing F1 with a double recessive parent wherein it's a 1:1 ratio of tallness to dwarfness, then you know that the genotype of the F1 generation was heterozygous. Because if it was homozygous, then you would get all of them being tall, correct? So if you change this small t here, if the F1 generation was a homozygous tall, then this one would be capital T here. So all of them would end up being tall, and you would not get a 1:1 ratio anymore. And so only when you get a 1:1 ratio of half of them being tall and half of them being dwarf, you know that the F1 generation was heterozygous and not homozygous. So the next thing we'll be talking about is incomplete dominance. Incomplete dominance is also known as partial dominance or blended inheritance, and it was something that was proposed by Carl Correns in 1903. Now, incomplete dominance is very different from Mendelian genetics because it's going to say that there is no dominant and recessive gene. Okay. The thing with incomplete dominance is that the dominant gene, or actually one of the alleles of the allelic pair, is not going to express properly. And more times than not, it's the dominant gene. So the dominant gene is not going to express completely, and therefore it is going to give rise to an intermediate phenotype. So rather than having a dominant phenotype or a recessive phenotype, you will have an intermediate phenotype. An example for that is Mirabilis jalapa, which is known as the 4 o'clock plant. Now it has two kinds of flowers, the dominant being the red and the recessive being the white. Now if you take a pure breeding dominant homozygous red flowering plant and cross it with a double recessive pure breeding white flowering plant, F1 generation you should get a red flowering plant in case of Mendelian genetics, what we see in monohybrid cross. But in this case you will get a F1 generation where the flowers are pink in color, but the genotype is like that of F1 generation that you would find in monohybrid cross. So although it is a heterozygous genotype and it should be the color red, but because the dominant gene is an incomplete gene, meaning that it is not going to express completely, so you will have an intermediate color between red and white, which will be pink. But if you take the F1 generation and cross it, so you are crossing the heterozygous with heterozygous again, F2 generation you will get the same phenotypic and genotypic ratio like that of any F2 generation in case of monohybrid cross that we were talking about in Mendelian genetics, where you will get a homozygous red dominant, and then you have the F1 generation incomplete dominant 2, and then you will have the homozygous recessive white. Okay, so this is with incomplete dominance. So the dominant gene is going to be dominant only when it is homozygous. If it becomes heterozygous, it is going to become incomplete dominant, and therefore you will have a phenotype which is in between the dominant and the recessive. If you are crossing the F1 generation with the F1 generation again, you can of course get the pure breeding dominant red flower plants, and you can get the double recessive white color plants. Okay. Another example for incomplete dominance is P starch grain size, and the starch grain size in P is controlled by the gene B. Now, because it is incomplete dominance, if it is a homozygous dominant gene, then you have round large seeds. If you have homozygous recessive, then you have wrinkled small seeds. If you have heterozygous, because it is incomplete dominance, the dominant gene is not going to express completely. So instead of having round and large seeds, you're going to have round and small seeds. Okay, and like in Mirabilis jalapa, if you're, this is what you will get in the F1 generation. If you're going to cross F1 with F1 generation, you will be able to get back the double homozygous dominant and the homozygous recessive. Okay. Other two examples for incomplete dominance is snapdragon, which is a flowering plant. Scientific name of it is Antirrhinum maius, and like Mirabilis jalapa, it has red and white flowers as the dominant and the recessive, and then in the F1 generation you have pink flowers because the dominant gene is not completely dominant. And another example is the Andalusian fowls, and they have feather color as black, which is going to be only there when it is a homozygous dominant character. It is going to have white color feathers when it is going to be double recessive, and it's going to have blue colored feathers when it is going to be heterozygous in nature, which will come in F1 generation. And like the other two examples that I was talking about, if you're going to take the F1 generation of either of them and self-cross it, you will be able to get back the homozygous parents. So the next thing that we'll be talking about would be codominance. Now, unlike incomplete dominance, codominance is a variant wherein both of the genes are equally dominant. So there is no recessive gene, there is no dominant gene. Both the genes act like dominant genes. So both of them get expressed at the same frequency. So there is no difference in how much of expression is taking place. Both of them are getting expressed. Both of them act like dominant genes. Now, an example for codominance is the ABO blood grouping in humans, which is controlled by gene I. So if you have just gene IA, so if you have IAAA, then you will have blood group A. In case you have only gene IB, then you have blood group B. But if you have a mixture of gene IA and gene IB, then you will have the blood group AB, because both IA and IB are getting expressed equally. So both of them act like dominant genes. So that is a major difference between codominance and incomplete dominance, is that in incomplete dominance there is a dominant gene and a recessive gene, just that the dominant gene is not getting expressed completely. But in case of codominance, you don't have a dominant gene or a recessive gene. Both the genes act like dominant genes and they both get expressed at the same rate, and so you have phenotypically both of them being present. So in case of incomplete dominance, phenotypically you will have a an expression which is in between the dominant and the recessive. So in case of snapdragon or 4 o'clock plant, you have pink color which is in between red and white. But in case of codominance, it is not in between anything, it is both of them being expressed, and so it is an expression, phenotypic expression, which is unlike both the parents. So it will not be like A, it will not be like B, it is something completely different, which would be AB.